So today we are going to play with fire, is it? No, you're not going to play with any fire. This experiment is also a simple experiment where you have to stain the nucleic acid and it goes like this. So let's go ahead, let's see how many things do you require for this. So over here you would be requiring the onion peel. You might get it exactly like this, or probably you might have to take it up. And my dear children, you would require a watch glass containing water, because after taking the peel we are going to put it inside the watch glass so that it does not gets dried up. Post that we will also require the glass slide as usual. Onto this glass slide you are going to transfer the peel and you are going to put Aceto-carmine stain onto it. Okay. And my dear children, after that you can put the cover slip, dab off any extra stain which is there with the help of a blotting paper, and then you would have to use the spirit lamp for heating it up for only 10 to 15 seconds. Now for this you will require a holder so that you can hold the slide as well. Okay, and then we have to view it under the compound microscope and make sure you view it only under 10x. Let's go ahead. So my dear children, today we'll need onion peel. Slowly try to peel off the innermost skin and then put it in a watch glass containing water. This is to make sure that it does not dry up. Further, with the help of a brush, transfer it onto a clean glass slide and make sure that there is no folding. And then you can use a dropper to put a drop of aceto-carmine and further warm it up over the spirit lamp for just 10 to 15 seconds. You can put a drop of aceto-carmine again and then later put the cover slip and further view it under the microscope and make sure it's under 10x power. And ta-da! We did this experiment as well. And my dear children, you would be observing something like this under the microscope. So as you can see, there are stacks of cells which are arranged very close to each other. And this red or pink color thing that you're able to observe is nothing but your nucleus. And what did you do to make your nucleus glow like this? You used a stain which is known as aceto-carmine. This whole particular experiment is on nucleic acid staining, right? And nucleic acid such as DNA, RNA, everything is found inside what? Your nucleus, and you wanted to stain that. Okay, your DNA is negatively charged and I'll say it is acidic in nature. So for staining this up, you need a basic dye, and that basic dye is what? Aceto-carmine. You know, positive, negative, they attract, which is why aceto-carmine is able to stain this nucleic acid up, and we were able to see this under the microscope. So let's see what are the general steps that have to be followed according to the lab manual which is given to you, which is uniform to all of you. Now obviously there are certain steps which you do differently in your institution. Please do it in the way your teacher have told you itself. Okay, now coming over here, let's see the general steps. First, peel off the epidermis from the scaly leaf of an onion bulb. So first thing, we have to peel it off, the epidermis from the scaly leaf, and then what we have to do, we have to place it on the slide and we have to add few drops of water. Why? Because water prevents drying up. Okay, water prevents drying up. Now take out a small piece of the epidermal peel. Now suppose over here in this particular video it was already small, so you do not need to cut again. But suppose you are getting a bigger peel, so you have to make sure that you cut out only a small peel out of it. Wipe out the water carefully using the blotting paper out there, and then you have to add 2 drops of aceto-carmine. Again, you can use a dropper. In this particular video we already had a dropper like thing at the top, which is why I used that. Now over the epidermal peel and heat gently on a spirit lamp. So you have to heat it gently on a spirit lamp for almost 10 to 15 seconds. That's all. Why are you heating it up? Because you want to soften the tissue. You want to soften the cells so that it takes up this particular stain, that is aceto-carmine. Okay, it's a plant cell, and plant cell has cell wall, a lot of things. So you want to make it soft so that it can take things up. Now, apply a cover slip over the epidermal peel, avoiding air bubbles, and wipe out the excess stain with the help of a blotting paper. Yeah, and then observe the prepared slide under the microscope. Now, this will give you 3 marks for sure. Again, you will need 2 marks more for getting 5 marks. And how will you get that? You have to do 2 things. First is reporting the shape of the cell. So we say the shape of the cell is what? Rectangular. Even though it's not exactly rectangular, but we say it is almost rectangular in shape. So please write this. Now reporting the number of nuclei per cell. So as you can see, in this particular cell you just have one. Similarly, in another cell you just have one. What do you call this condition as? You call this condition as uninucleate condition. Uninucleate condition. If you write this, you get 1 marks. If you write this, you get 1 marks. 1 plus 1, 2, and 2 plus 3, 5. You have got your 5 marks in this experiment as well. Got it? It's that simple. Now, one very important thing. Have you subscribed to the channel yet? If not, Baba, subscribe to it right now. Because ultimately, we will definitely help you in 110 ways possible by us. So please trust us and keep on trusting us if you already do, and lots of love from all of us. Okay, now one very important thing, my dear children. So in the first question, as I told you, you had 4 experiments. You need not do all the 4 experiments. According to the lottery system, you would pick a chit, and whatever experiment you have got, you would have to do it. So I have already done the video on pollen germination, pollen tube growth, as well as the TS of ovary. If at all you have not watched these videos, go watch this, because by luck you might get anything out there. So do not take any risk. And also over here we have done the nucleic acid staining experiment. With that we end up all the first question, all the questions which were there under category 1, and all of them will give you what? 5 marks. Okay, now we will be going ahead with the next question, which is common to all of you. Everyone has to perform this experiment, and that is the experiment on mitosis. Now this experiment, people think it's very difficult. Trust me, it's not too difficult. Okay, and PU2 board has made it a bit more easier for you, and I'll be talking about that in the next video, so stay tuned for that. And also, if at all you are someone who is preparing for KCET examination, go check out our ABHYAS KCET test series. It has 35 mock tests, and there will be chapter wise KCET PYQs. This is something that we have added up newly, and there will be questions along with some proper timing. So it's like exam like conditions will be maintained so that you have an understanding of your speed and accuracy. If at all you want to know more about this, definitely check it out in the link given in the description section. And my dear children, it's one of the most trusted test series out there, and since you guys asked it, we launched it a bit more earlier this time. Okay, so check it out if at all you think you need it. And that's all for today. Wishing you all the best. Lots of love from me. Bye bye. Take care.